Hi, I'm Allison Gregory from Oregon Health and Science University, and I'm going to present some data that we've generated from the PLAN Ready Natural History Study. Specifically, it's focused on the infantile version of PLAN, also called INAD, and this was recently accepted for publication, so you're getting a little preview of something that should be out in the world soon. So for those of you who aren't familiar, this is a 5-year study that parents typically complete for their child with INAD, and it's intended to look at the natural history of the disease over those 5 years, understanding how typical development happens, how they start to lose skills, what the main symptoms look like, what the progression is, and um, what we can measure across that spectrum. So the aims of the study are to look at the age of developmental milestone loss, meaning is there a certain pattern to when they lose typical milestones like sitting up on their own, rolling over, making certain babbling sounds or single words, doing things like feeding themselves, etc. Tracking the age of disease milestone acquisition. A disease milestone would be a common symptom that we think happens in INAD, something like optic atrophy, seizures, getting a gastrostomy tube, those sorts of milestones. And then finally, assessing health quality of life using a tool called the Infant and Toddler Quality of Life Scale. So this is just an overview of what our study population looks like. This was a group of 40 participants or kids with INAD, roughly half female and half male. We got about as close as we could with that at 21 and 19. Their age at enrollment on average was 5 years, and they're from a number of different countries, but one of the requirements was that they be fluent enough in English to complete the study. So this bar graph shows the number of visits on the x-axis, the number of participants on the y-axis, and it gives you an overview of how many visits have roughly been completed by the different participants. So because enrollment is rolling, not everybody joins at the same time. You know, some are just getting started this month and some have been doing this for 5 years. So when you look on the left side of the graph, you know, there are 6 individuals who have only done 1 or 2 visits. That's maybe, you know, half a year, while there are 2 on the far right who have completed 17 to 20 visits, which is essentially going through the whole duration of the trial. And then a lot of people in the middle who are sort of partway through. So the first thing that we did was look at these developmental milestones, and we generated these histograms, which are shown on the screen. The histogram, each one individually, you know, has age across the x-axis, the number of participants on the y. The gray background shows you the total number of data entries we had for each age. So that's why the gray background, the shape of it and the pattern never changes. It's always the same. What changes by milestone are the dark black bars, which show you how many of the kids had that milestone at that age. So, for example, we only had a few 1 to 2-year-olds on the far left there. That's the dark gray. We only had one 17-year-old on the far right. It's sort of a bell-shaped curve where you can see in the middle is where we have the most data points. And the other thing that I should mention is an individual child can be represented multiple times across one of these histograms because they keep doing entries every 3 to 6 months. And that's okay. So what it's showing us is, you know, it's like a snapshot at age 5 or at age 9 of how many kids with INAD have this milestone. When you look at these, you can see, for example, with vocalizes, that the dark bars almost completely overlie the light gray, and that means that all those kids can vocalize across the whole disease. Whereas if you look at something like picks up small objects on the bottom right, that's attainable for some kids early on from about age 2.5 to 7. But beyond that, it's uniformly not happening. And before that, you know, it's maybe a third who have been able to reach that, or in some cases up to about a half. So here are some more of those histograms, just giving you an impression of sort of what skills are more common, what ones maybe don't happen at all or only to a certain age. And we've drawn some conclusions about that. So basic language milestones are more likely to be attained in the early study visits. Fine and gross motor skills, what we noticed was a tapering down by about age 8, where a lot of those skills are happening up until age 8. Maybe there's some gain and loss, but then they're quite uncommon after that age. And then engagement, oops, sorry. By engagement, what I mean is a child engaging with their environment, with their family through laughing, smiling, vocalizing, and it was heartening to see that happening pretty uniformly across the entire disease span up to the older ages. And I think that's an important sort of gem from a genetic counseling standpoint, is to be able to tell newly diagnosed families that kids do continue to engage with their families in that way. The other thing I should have mentioned with the fine and gross motor skills more specifically is that that pattern up to age 8 really applied mainly for picking up small objects, pulling to a stand, sitting on their own and rolling over, while walking independently was actually quite rare at any age. So the next thing that we looked at were what we call disease milestones, which I explained previously. And these types of graphs are called survival curves. The age, again, is on the x-axis. The y-axis is what's called the survival probability. And the way to think of this is sort of survival of that milestone. It's the name for this kind of analysis. But it always starts with everybody not yet having attained that milestone, the disease symptom. And then as time progresses, you'll start to see the line moving down and to the right as some children gain that symptom. So you'll see, it goes from nobody having it to 25% of them having it and so forth. And when you look at these, if you look, for example, at panel H or I, those ones are pretty clean. There's just one line there. That's because for something like a tracheostomy, when the children entered the study, none of them ever had a tracheostomy at the time of enrollment. It's something that came later for a few of them. So that line is pretty flat because only a few obtained it. If you look at the graph, it's usually around between age 5 to 8 or so that happened. Similarly for death, obviously they were all living at the time of enrollment. We did have some deaths over the course of the study where we were starting to be able to track that along that survival probability on the y-axis by about 50 by the median. So, half of those who died had done it before age 10 and half about after. The other thing to point out here briefly, but I don't want to focus too much on it, is that the dark lines for all of these graphs represent children who did not have that milestone at enrollment. However, because some kids are diagnosed late and they've already reached some of these symptoms or disease milestones, we had to create a separate line for those and keep them sort of separate. So the light gray line reflects for that disease milestone all the kids who already had it when they enrolled. So something like looking at optic atrophy or seizures or gastrostomy, these are all GE reflux. These are good examples of what I'm talking about, where there's a gray line sort of shifted to the left. What's interesting about that to us is, we didn't know. We tried for medical records and, thought about how we might collect information retrospectively. So things that happened before enrollment to know when these kids started having some of these symptoms. We didn't have a good uniform way of doing that. So instead, we sort of cheated, and the gray line represents the age when they enrolled in the study. So they could have actually attained the milestone several months before that. We don't know. We just kind of tried to roughly guess by doing age at enrollment. And what's interesting to us is that line is still shifted quite a bit to the left, meaning they appear to have had the milestone much earlier than the group that didn't have it at enrollment. And we have some thoughts about what might explain that I'll go into with the next slide. So these results, 4 of them had tracheostomies over the course of the study at a median age of 6.2 years. 8 died at a median age of 10 years, meaning half were above 10, half were below. And then, of course, 32 were still living. I mentioned these disease milestones before with the light and the dark line. And the other thing that we've speculated is, why would that light gray line be shifted to the left? Why would those kids have these milestones earlier? We suspect that maybe on the spectrum of INAD, those are the ones who are more severe, that had more severe disease to begin with, which would probably mean earlier onset and more rapid progression and attainment of those things we were measuring. So that probably is reflected in that shift to the left. And if we knew the true age when they reached each of those milestones, I suspect it would shift it even more to the left. So then finally, we also looked at this infant and toddler quality of life scale that looks at health quality of life. This is a scale that's used in kids who are 2 months old to 71 months of age, which is just right up to 6.0 years. And we did have available to us what's called a normative population from the United States of like-aged children, meaning, those who are quote unquote normal, to compare with our INAD group. And you can see, so in the first column there, this is a scale that measures things like overall health, physical abilities, growth and development, body pain and discomfort, temperament and mood, some measures of different behavior, impact on parents, both emotional and in terms of their time, and also family cohesion. So the maximum score is 100. So just as an example, if we look, for example, at physical abilities, the PLAN kids had an average score of 21 on a scale of 100. The US comparative normal population had a score of 96. That's quite a huge difference. That gave us a p-value of less than 0.001, which statistically indicates that that's not, by chance alone, that it is a real difference. So if you keep going down that p-value column, most of those categories we saw a significant difference. The other thing we did with this quality of life scale was look just within the kids with INAD to see if their scores changed over time. For example, do their parents, score them differently at age 2 than age 5, and why might that be? And we did see several differences. So again, this time, if you look at the second column here, we were able to calculate the annual change in their standardized score, meaning from year to year, does the score go up or down a certain amount overall when we lump everybody together? And the answer was yes. For some categories, it does that in a way that we think is, again, statistically significant that we can prove. So for example, physical abilities, they tend to be rated annually with a 6-point loss, which makes sense because we know that with this disease, kids lose abilities over time as their disease progresses. So we saw, similar trends like that, significant differences in these areas that I've circled in red, which is several of them. And you can imagine that when we think about doing an interventional trial, like a clinical trial of gene therapy, that having this data set can be very valuable because other investigators and people developing those therapies can use it as their control group or as their comparator group. So they can look at our data in our 40 kids, they can look at their own 40 and see if their intervention changes the trajectory of any of these things, if it changes the way the parents rate them, if it changes the time when a certain milestone is attained or the pattern of that. So that's part of why we're doing this work. The other thing I should point out that I didn't mention at the beginning is the way the study is designed, it's considered to be prospective, which is sort of the gold standard from the FDA's standpoint. So when you collect something prospectively, you start measuring it at one point in time and you just keep marching along, collecting it as it happens in real time. And because you're doing it that way, you can be very consistent. You can use the same tools and scoring methods with everybody involved. And you can, be, I think, a little more confident in some of the quality of your data. The downside is we don't know what happened before they sort of walked in our door, which gets back to that other problem I mentioned of not knowing when milestones were attained prior to them enrolling. So results from using this quality of life tool, there were significant differences in all categories. The greatest impairments overall were in physical abilities, growth and development, general health, and parental emotional impact. We also saw the greatest annual change in physical abilities and changes in behavior. And overall, what we've concluded is that, as we aspired to do over 5 years ago, we've generated key reference standards that can be used by other investigators and in interventional trials. We have generated some valuable information from a genetic counseling standpoint in terms of being able to tell families more about what to expect, and some of those encouraging, tidbits about how kids will continue to interact with their families long term. And then finally, limitations. I just mentioned the biggest one, I think, which is, we do have a limit in lacking retrospective data, and we need to think a little bit harder about maybe how to incorporate that. And that is it for this section of my talk. Thanks for listening.
Good morning to everyone. My name is Ivano, and I am a senior scientist at the Neurological Institute Vesta in Milan, Italy. And today I would like to tell you something about our ongoing project on PILAN, which is called PILAN-MARC. So PILAN is a rare genetic neurodegenerative disorder which is caused by mutations in PLA2G6 gene. PILAN is associated with some typical clinical signs, such as movement disorder, developmental delay, cognitive decline, speech problems, behavioral changes, and high mortality risk. Based on the onset of the disease, we can divide the pathology into 3 subgroups, depending on the onset. There is the infantile onset with the INAD, the atypical juvenile onset, and the late onset associated with Parkinson's. In this cartoon, I would like to remember the very long and hard journey about the PILAN translational research, which started several years ago with the identification of the gene associated with the disease. Since then, several groups started to study this pathology, achieving many results. For example, the description of the protein function. Then several models have been generated, both animal but also cellular models. And studying these models, we were able to identify some of the pathological mechanisms associated with human diseases. For example, the mitochondrial dysfunction, the lipid alteration, the oxidative stress, and also the iron dyshomeostasis. All these mechanisms can be used as therapeutic targets to design specific clinical therapeutic approaches. And some groups designed dedicated therapeutic approaches which have been tested on the available models generated so far. So we are now at this point in which we have some promising approaches that can be directly translated to the clinical practice through the design of a dedicated clinical trial. However, the real question today is, are we really ready for therapy, for PILAN? This is a rhetoric question because, of course, we must be ready after all this time. But in order to obtain the best of the results from a future clinical trial, we have to address some open questions, some issues. One of these is linked to the outcome measures that are the measurement that we must provide during a clinical trial in order to demonstrate that the therapeutic approach we are testing is effective in modify the disease. Today, for PILAN and also for many other types of neurodegenerative disorder, we only have outcome measures linked to the clinical evaluation. For example, the use of neurological scales, but also the MRI imaging and the assessment of the home life of the patients. However, most of these outcome measures can be biased, or they are non or semi-quantitative, and for sure they can have slow responsiveness. Remember that PILAN is a progressive disease, and sometimes this progression can be very slow, and probably also the response to a therapeutic approach can be slow. So the question is how to measure the effect of a treatment in a fast and reliable way. Remember that today many clinical trials for progressive disorders fail because of the lack of objective and reliable measures. Second, the traditional medicine, in the traditional medicine, we have a therapeutic approach that we apply to all the patients of a given disease. And we can have a group of patients that respond to the therapy in which the therapeutic approach is effective, but also a group in which the therapeutic approach is not effective. And a small group, hopefully a small group of individuals in which the therapy could be also detrimental. So we are going today through a new kind of medicine, which is called precision medicine, which is based on a more personalized diagnostics and therapeutic approaches. This is based on the analysis of samples from patients in order to stratify the patients in a different way based on molecular data in order to apply different form of therapies or modified form of the same therapy in order to maximize results. So to overcome these issues, we need biomarkers. A biomarker is a biological marker, is a characteristic objectively measured and evaluated that indicates normal biological process, but also can give us a response to an intervention, including the therapeutic approach. There are many examples of biomarkers, and the ideal biomarker should possess at least 4 characteristics. First, its expression must be significantly altered in the related disease condition, for example, in our case, in PILAN. Second, its expression must correlate with the progression of the disease, which this means that the changing in the biomarker should correlate with the changing in the progression of the disease. Third, the biomarker should be readily quantifiable in an easily accessible clinical sample, for example, blood, urine, saliva, etc. And also, of course, it should be economical, quick, and consistent. So, as I told you before, today we are some therapeutic approaches preclinical therapeutic approaches that are very promising. One of, above all, there is the gene therapy approach. However, today, no reliable biomarkers are so far available, and this is difficult to quantitatively assess the impact of a therapeutic approach on patients during clinical trials. So we aim with our project to find potential circulating biomarkers which are able to advance our understanding of PILAN, but also to help the so-called therapeutic readiness. So this is our project, which is designed in a time frame of 2 years. The first year is dedicated to the clinical part, and the second year is dedicated to the laboratory analysis. So we are now at this point. So we already obtained all the ethical and regulatory approvals, and now we are enrolling a small group of patients, but also healthy controls. Patients are clinical examined by using the new PLAT-UG6 associated neurodegeneration rating scale, which has been recently proposed by an international group of clinicians. And during this examination, there is the collection of different types of samples, such as blood, plasma, and cerebrospinal fluids, which will be then, during the second part of the project, will be analyzed in laboratory at different level of analysis, such as transcriptomics, targeted proteomics, circulating microRNA, and circulating mitochondrial RNA. At the end, all the data obtained by this analysis will be integrated with the clinical data in order to find potential biomarkers. So to give you more details about our analysis, the first level of analysis will be bulk transcriptomics. This means that we will use next generation sequencing approaches in order to analyze, to quantify the expression of all the transcripts in the whole blood. We are talking about more or less 20,000 of genes in order to find those genes that are differentially expressed in patients compared to healthy control, in order to identify a molecular signature. The second level of analysis will be the quantification of the circulating microRNA. MicroRNA are small non-coding RNA molecules that are able to regulate the expression of many genes. And they are very promising into the field of biomarker discovery because they are actively secreted by the cells into body fluids, and also they are very stable in circulation. And today we know that many different microRNA are associated with different types of human disorders, including neurodegenerative disorders. So we will analyze simultaneously the amount of circulating microRNA, more than 700 of microRNA, in order to find specific microRNA which can be altered in patients against controls. The third level of analysis is a targeted proteomics. So we will use for this purpose a new approach, which is the new LISA platform, which is a recent approach that combines a proteomic approach and also a next generation sequencing, providing an ultra high sensitivity and scalable multiplexing assay. So we will analyze 120 proteins, which are mainly involved into inflammation and neurodegeneration, which are the two characteristics of PLAN, with a very high sensitivity and also high reproducibility. The last level of analysis will be the quantification of the circulating cell-free mitochondrial DNA. So mitochondria are the powerhouse of the cells, and they also possess their own DNA molecule, which is released by mitochondria and also by the cells after some stress circumstances. For example, oxidative stress, but also neurodegeneration, neuroinflammation, and mitochondrial dysfunction. And we know that in PLAN there is a mitochondrial dysfunction. So it has been demonstrated that in some human disorders, the levels of the mitochondrial DNA in body fluids is increased. So with our analysis, we will be able to analyze the exact number of mtDNA copy in the body fluids in order to find if there is a correlation between the number. There is an increase in the number of circulating mitochondrial DNA in patients versus controls. So at the end of the project, all the data is very big amount of data will be integrated also with the clinical data by statistical and bioinformatic approaches in order to find, of course, candidate biomarkers, which will allow us to perform a patient stratification and also to identify possible new pathway that are involved into the pathogenesis of the disease, which can be used as therapeutic targets to design new therapeutic approaches. So in summary, our project will possibly identify potential circulating biomarkers in a small cohort of patients by integrating molecular and clinical data, and potential biomarkers will be validated on a larger cohort in a future and bigger project with the involvement maybe of many other groups in the world, hopefully under the umbrella of the NBIA Alliance. So to conclude, I want to thank all the people which are involved, that are involved into this project. Our group in Milan at the Vesta Institute, especially Dr. Giovanna Zorzi, who is the director of the infantile neuropsychiatric unit, Valeria Tiranti, which is the head of our laboratory, and Chiara Cavestro, who is a PhD student mainly involved into the laboratory analysis. And of course, a special thanks to the NBIA Disorder Association and INAD Warriors for their economical support. And thank you for the attention.
Good morning, everyone. Thank you, Patty, Amber, and the rest of the NBIDA for inviting us to present on our gene therapy program today. I'm Lena Panwala. As many of you know, my husband and I started the INADcure Foundation in 2017 after our daughter Aria was diagnosed. Like most parents, we wanted to do everything possible to help her and other children like her. So we joined forces with our families and their communities to raise the funds needed to support early research efforts at Washington University and Baylor College of Medicine. Those early studies led us to gene therapy, which currently represents the most promising path forward with the hope of targeting the root cause of INAD and the potential to improve the quality of life for our children. Gene therapy works by using an AAV vector to deliver a healthy copy of the PLA2G6 gene directly into the cells. The goal is to restore enzyme activity, slow or even stop disease progression, and make a meaningful difference in how our kids live and grow. Because INAD is so rare, getting the attention of biotech was challenging. So the INADcure Foundation made the decision to step up and lead the effort ourselves. We became the official sponsor of the program, raising the necessary funds, bringing together an incredible team of scientists, doctors, and experts, and staying involved every step of the way to keep things moving forward. Thanks to generous donors, grants, and tireless grassroots fundraising, we've made incredible progress. And today, I'm excited to share for the very first time that we'll begin manufacturing our gene therapy this August. This is a huge milestone for the INAD community, and it sets the stage for the very first human trials, which we hope will begin in early 2026. You'll be hearing more about this exciting progress later this hour from Dr. Neil Hackett and Dr. Darius Adams. But first, I'm happy to introduce Jennifer San Miguel, Senior Study Director at the Rare Disease Translational Center at the Jackson Laboratory. Jennifer will be talking more about the preclinical studies that helped us get closer to making this treatment possible. Thank you.
Thank you, Lena. And everybody can see the slides okay in the room? I just want to double check. Yes, okay, great. Thank you. All right, good morning, everybody. My name is Jennifer San Miguel. I work at the Rare Disease Translational Center as the lead scientist working with Lena and her INADcure team. And I'll give some brief updates on the preclinical work that we've been doing. So we have a very faithful mouse model of INAD, and what I'm showing is just a representative graph of what expression of the PLA2G6 gene looks like in our wild-type mice compared to our INAD mice that have 2 copies of a mutated gene. So our mice have severely reduced expression, which mimics what we see in patients. And in our mice, this leads to nerve cells getting sick and degenerating and dying. The mice develop tremors. Eventually, they'll start losing weight. They'll start losing their ability to walk or right themselves when they're on their sides. And this ultimately leads to a much shorter lifespan in these homozygous animals. Our proof of concept study ran last year, and I want to give some updates on our dosing study that we just completed. The goal of this study was to treat this faithful model in mice of INAD with 2 different doses of our AAV9 gene therapy to determine if we could measure efficacy, aka improvement in the phenotypes of these mice, such as survival, body weight, and overall motor function. So we injected our newborn mice into the brain, directly into the cerebrospinal fluid with either a control, so there was no gene therapy, a high dose or a low dose of the gene therapy. And we treated either wild-type mice who have healthy normal function of the gene or homozygous mice who carry the 2 copies of the mutant PLA2G6 and are the disease model. We monitored a number of aspects that I just mentioned across a 40-week study period. And I'll just show 2 key pieces of data from this study now. One of the things we wanted to assess was motor function in our animals. So we developed a score based on a scoring system that was developed for ALS mice. This involves hind limb extension. So if you pick up an animal by its tail, the normal response of a mouse is to spread out its legs because it has the muscle strength to hold them up. And we assess if they can hold up the legs or if they are not strong enough and they have to drop them down. We also assess their ability to walk. We look for things like a normal mouse is able to pick up its feet and ambulate, and a mouse that's having a hard time with their muscles or nerves are going to drag their feet or drag their limbs. We also assess if there's any signs of paralysis in any of the limbs, and if they're able to right themselves when we put the mouse on its side, is it able to get back up on its feet within 30 seconds on either side. So this is a semi-quantitative scale. If there was no phenotype, it was scored as a 0 or 1 if the phenotype was present, and then we added those all up. So what I'm showing is these scores for males on the left and females on the right. Our wild type controls are these orange lines on either side, and you can see on the scale they're only reaching a 1 pretty much on average. So a low score means a healthier motor phenotype. You can see the males that were not treated with any gene therapy got very quickly very severe scores, and unfortunately, these animals did not live long enough to collect any more data in this study. The purple or maroon dose, I should say, are the low dose treated animals. You can see their severity scores increase more slowly over time, and then the high dose increase even more slowly. So this means their motor function is improved for longer in a dose-dependent manner. And we see the same thing in the females. These are the mice that were not treated with gene therapy in the red line. So their scores got worse more quickly than the low-dose animals compared to the high-dose animals. So this is a very encouraging result. The last piece of data I'll share with you is the survival curves. So again, we're looking at males on the left, females on the right, and we're looking at probability of survival. So 100% means everybody is alive. And then we're looking across age and weeks of the mice across time. You can see the untreated homozygous animals, males and females in the red, succumb to their disease more quickly, with all of the mice passing away by 25 or 30 weeks respectively. And what we see is a dose-dependent increase in survival in both the males for the low dose and even better survival in the high dose. And the same thing in the females. There's an improvement in survival in the low dose and improvement in survival even more in the high dose. These results were statistically significant. I'm showing you p-values. What that means is what is the chance that we would expect to see this result by chance. And when you have a very, very, very small number, that means that result is almost impossible just due to random chance. So this really strongly suggests that gene therapy is having a very significant effect. So overall, we have a faithful model of INAD in our mice. We were able to treat with 2 different doses of our gene therapy, and we observed improved motor function scores and overall survival. And with that, I will turn it over to Neil.
Okay, good. I'm Neil Hackett. I'm a research professor at Weill Cornell Medicine in New York, but more importantly, an advisor to INADcure. And I want to go through quickly the process of how you go from the study we've just seen in treating mice with INAD to getting a clinical trial. And this all hinges on the Food and Drug Administration. And INADcure engaged them early and has received 2 items of recognition that are important: the orphan drug designation and the rare pediatric disease designation, which basically recognizes the potential legitimacy of the treatment and the severity of the disease that we're trying to treat, which gives you some special considerations with the FDA. Our next interaction, targeted for the 3rd quarter of this year, is to submit a document called the Investigational New Drug Application. This is a very complicated document, gets up to 1,000 pages very often, but it's got 4 basic chapters. The first one is the efficacy data, which we've just seen, basically showing that this treatment, this gene therapy treatment, has a strong prospect of being beneficial, perhaps very beneficial to children with INAD. But you also need 3 other elements. You need a manufacturing plan. You need to show that the drug is safe, even at high doses. And you have to have a clinical trial design. I'm going to go through the manufacturing and toxicology very simply, and then pass it on to Dr. Adams, who will talk about the clinical aspects. So manufacturing is complicated. You have to make sure that the drug you're making is absolutely pure and is exactly the same every time you make it and is always potent and effective. So the FDA has this standard called good manufacturing practice, and it requires drug production to be done in a clean room facility. The technicians have to get into these bunny suits and keep everything immaculately clean. You have to identify pure reagents for every item that you're going to put into the drug. You do this all in a extremely sterile clean room facility. And after you've made the drug, you have to do all kinds of purity testing. And there are a lot of complicated aspects to it. So, for example, if your technician has had influenza, they cannot go back into the facility for a week because there's always a chance of contaminating the product. So it's a rigorous control, but it ensures that the quality of the product is extremely high and that the product is safe. The next step you have to go through is toxicology. This is done primarily to show that the dose that you're going to use in humans is almost certainly going to be safe. So the mouse study that you've just heard about, that told us what dose we needed to get a fairly dramatic increase in neurological status in the mouse. And that tells us what dose we're going to try and use in humans. You scale it up on the basis of body weight. And of course, a mouse is 1,000th of the body weight of a human, but you still have an estimate of what dose you want to do in humans. But then you want to be sure that that dose is safe. So what we have done is we've taken a rats and given them various doses up to 10 times the planned human dose. And therefore, since those rats were healthy, even when we gave them a very high dose, we believe we have at least a 10-fold margin of error of safety when we go into the human study. So summarizing the overall status, the efficacy is complete. As Lena has just told you, the manufacturing starts in August of this year, and that will take about 4 months to complete. The toxicology is complete. The clinical study is in advanced planning, and we're intending to do the FDA submission in the late third quarter of 2025 so that we can get the clinical trial started in 2026. And with that, I'm going to hand over to Dr. Adams.
All right, thank you, Neil. Let me just get my, here we go. Okay, so yeah, I just wanted to chat a little bit about the clinical trial components. Once we get through all of the basic science studies, it's important that we go through the process in order to test this first in human studies. And really the goal here is to establish safety. Let's see if I can advance my slides. There we go. So really the goals here are primarily to establish safety initially, but then we also look at efficacy early on. Clinical trials are performed in phases. We start with a small group initially to establish safety, and if the treatment appears generally safe, we'll proceed to efficacy analysis. However, in rare disease trials, safety and efficacy assessment is commonly combined. So this is sort of the standard workflow, phases 1 through 4. However, if we are able to demonstrate efficacy even early on, that could get us approval from the FDA. So we're planning on doing a phase 1/2 clinical trial. So in the US, FDA approval is needed prior to initiating clinical trial, as Neil outlined. They review overall study plans, offer guidance, and then they review data and results from each phase of the clinical trial. And as I said, in some cases, they will approve treatment if pretty dramatic success is noted early in the process. But this is why we have ongoing interactions and discussions. So as part of the clinical trial, we have inclusion criteria. This is standard in every clinical trial. And of course, we want to make sure we're selecting individuals who are appropriate for the trial. So for example, since we're targeting PLA2G6, one of the key parameters to be included in the trial is to have the biallelic pathogenic variants in PLA2G6. We do sometimes include, well, we always include other criteria. And so certainly having a condition other than neuroaxonal dystrophy would not obviously allow participation in the trial. So this is the draft clinical trial inclusion criteria that we have. And it just basically goes over, again, having PLA2G6 pathogenic variants, and just some other criteria that are pretty standard with clinical trials. A lot of it has to do with compliance and meeting age-appropriate criteria, for example, and being able to undergo anesthesia as needed in order to do the procedure. So for this clinical trial, it will take place in the USA. We will be presenting this to the FDA as a phase 1/2 clinical trial in order to assess both safety and efficacy. So we are planning, as mentioned, to go ahead and initiate the study early in 2026. But of course, that will depend on a variety of factors, including the response from the FDA. Okay, that's everything I have. Thank you.
Yep. Hi, I'm Heather Prentice. I'm with INAD Warriors. If it's going to be in the U.S., as we know, this is a really small cohort. What's your sample size that you're planning to have included?
So yeah, I think at this time the plan is to enroll 10 patients in the initial study.
Okay, so with that, I'm assuming there's not an age limitation, because that probably would be everybody in the U.S. It feels. I don't know what the actual known number of diagnosed children are in the U.S.
Yeah, so to clarify, it will take place in the U.S., but we will open it potentially worldwide. It's just that individuals from other locations will have to travel to the U.S. We are looking potentially at external collaborations, but for now, the goal is to at least have the infusion done in the U.S.
And then I think everyone's really interested in this, obviously, and wants to know, the clinical plan outlined. Obviously, money is a potential barrier. Can you help us understand the timeline implication based on where you are with funding and what you need to keep it moving forward, or if there's risk of the timeline adjusting because of that?
Oh, Lena, I think you're muted.
Yes, sorry about that. I had to step away real quick. Could you repeat that question one more time?
Yeah, just your clinical plan is pretty clear, but funding is an important element of that. Can you share with us all, based on where you are with that, if there's a risk of the timeline shifting out based on your current funding situation?
As far as funding is concerned, I think we're okay, but there's always risks of delays in timelines. There could be things outside of our control, whether there's delays from manufacturing aspect or things of that nature. But from a funding perspective, I think we're actually, on track. We should be able to keep this, program on track. Yeah. Mm-hmm.
Anybody else? Those are my questions.
Okay.
I wanted to ask, I noticed it was in newborn mice.
I'm so sorry, I can't hear the question. Can you use the same mic?
Mike's actually from the computer. Yeah. Oh, just go over here.
Yeah.
Can you hear me now?
Yes, thank you.
Hi, I'm Laureen Kearney. My daughter Sage has INAD. I know Lena.
I was asking, I saw that this was given to newborn mice and it had great results as far as extending the disease process and surviving longer. Is this something you think, just wondering if you feel like this is the, like you'll need multiple doses to continue to treat the patients since the mice, their survival curve was extended, but it wasn't like obviously normalized. And I guess related to that, how if you're the first 10 patients, I think more likely would be like newly diagnosed, or what the goal is with that. Thank you.
And maybe I can answer some of those questions. It's hard to generalize from the way mice are to the way humans are, and there's a level at which we really can't predict what we expect. The AAV gene therapy is always a one and done procedure, where the expectation based on other diseases is that the therapy lasts for many years, and there's some clinical approved AAV products that make this a reasonable assumption. Unfortunately, it can only be done once, but since it lasts for 10 years plus, that's not a limitation, I think. Ideally, we would do children in the distant future. We would do children immediately upon diagnosis. But as I understand the current clinical trial design, we're accepting a very wide range of ages. And until we've done the study, we really don't know what the impact would be as a function of age of administration.
This is Steve Kearney. So, I would agree that the mouse trial was striking and definitely dramatic. How related do you think it is to the clinical trial that you're proposing, where this is a wide variety of stages of disease? And, do you have a plan, or have you seen some data that shows that, intervention, maybe when mice start to become symptomatic, does have a similar effect?
We don't have that for INAD, but for other similar neurological diseases, there is data on age-dependent effective treatment. And it's certainly the impact is slightly higher as the mice get younger. But again, we have no idea to what extent that applies to our humans. Remember, the mouse brain is 1,000 times smaller than the human brain, and it's just not a comparable situation.
Hey guys, can you hear me? This is Allison Gregory. Yeah, hi. All of us in the room know that no gene therapy trial is without risk, but I'm wondering if maybe one of you can comment on what you're going to do to mitigate those risks for the families who are thinking about this in terms of like immune suppression, what the expectation is, how long you stay near a study site, these kinds of things that would impact their lives, short-term, long-term.
Yeah, so the current draft protocol does include immunosuppression, has been done with other gene therapy trials in order to mitigate any immune-mediated reactions. There have been side effects noted, and some very serious in other gene therapy trials, for example, in spinal muscular atrophy. So we're very aware of that and want to mitigate that as much as possible. So there will be, at least in the draft protocol, for the first 12 weeks, at least, weekly visits to monitor and do blood tests in order to make sure we're not having, for example, impacts on liver function and address that accordingly by adjusting immunosuppressive doses. So yeah, we're certainly aware that gene therapy is not benign in some rare cases, but based on the data, a lot of individuals who receive it can do well. But of course, we always want to consider safety first.
I have a follow-up on safety that might be helpful to the families to understand. A lot of trials will go from small animal models into larger animal models in toxicology, dogs, monkeys. You're not including that. Can you help them understand from a safety toxicology perspective the rationale of that?
Yeah, I can speak to that. In the FDA's, first of all, we take the FDA's guidance. The FDA have seen dozens of AAV gene therapy trials, and they know a whole lot more than any researcher or foundation knows. A few years ago, when you wanted to do AAV9 gene therapy, the FDA insisted that you did primate studies. But after they had seen a great many of those, they decided that generically AAV9 had an extremely well defined safety profile, and that they didn't need to see a whole lot of monkey study. And monkey studies are incredibly expensive and time consuming. And once the FDA was no longer insisting on them, people did not take that route. I think the safety profile of AAV9 is so well known that we know exactly what we're dealing with. And we have done a toxicology study to say that our gene does not do anything different from any other gene in AAV9 gene therapy. So the very extensive toxicology that a drug for tens of thousands of patients over many years would need is really no longer required for a rare disease with a pretty much known therapeutic entity.
Hi, I'm Stephanie. Hi, Lena. We're from Australia, so we are newly diagnosed this year, and it is something that my family would be interested in, as you know, Lena. I know Lena and I have spoken a lot about what that looks like for an international family if something was to go wrong in a trial. The US is very different to Australia, like how we would be covered in Australia medically. So I don't know if you have any insights into, if something was to go pear-shaped in the process, how would an international family be treated?
So yeah, so the protocol procedures, or at least part of the clinical trial, will be covered. But yes, that is a good point. If there is additional medical care that is required, it would be helpful to have potentially something like a travel insurance or something to give you some coverage for that. But that's something we're actively looking into to see what options are.